bring it down very far. Can you hear me? Good. So I'm going to talk about possible insights into pediatric skin disease from the Human Pediatric Skin Atlas. So I'm a pediatric dermatologist and researcher at QMUL. So I'm going to talk a little bit about the team, our research plan and questions. I'm going to talk about skin structure and cell types. I'm going to talk a little bit about pediatric skin diseases. And I'm going to talk about our approach to public engagement. So this is the team. So we've got Muz Hanifa at the Sanger. There you go, Muz. We've got Neil, Dave, who's here, and Bayan in Newcastle. And we've got me, Mirjana Evramova, and Enish Sakura, who's also here at QMUL. So for those of you who are not so familiar with the UK, now because of Brexit, things are very difficult, as you may hear in the news all the time. The Sanger is near Cambridge. Newcastle is up in the north of England, and I'm based in East London. I work in this institute, the Blizzard, and this is the hospital I work in, which is in Whitechapel in East London. So one important part of this project is diversity of ancestry. And the blue dots on this map represent the 3 hospitals of Barts Health NHS Trust. And basically we've got a lot of patients of South Asian Indian ancestry, which you can see in sort of dark red in one area. We've got a lot of patients of South Asian Bangladeshi origin immediately around my hospital and in the nearby boroughs. And we also have patients of West African and Afro-Caribbean origin. There's something called the index of diversity, and basically you've got a high diversity index if there's lots of different languages spoken. And the borough with the highest diversity index in the UK is Newham, which is quite near our hospital, and Tower Hamlets, which is immediately adjacent to the hospital, which is in red. So we plan to recruit 24 donors prospectively from European white ancestry and 40 donors from mainly South Asian and African, Afro-Caribbean ancestry. These will be children who are undergoing surgery for a different reason. So for example, they may be having a cyst removed from their back. So we will take the normal skin at the edge. We're going to look at children throughout the lifespan, so 0.3 months, 3 to 24 months, 2 to 7 years, 7 to 10 years, and 10 to 16 years. We're going to look at various anatomical sites, so trunk, limb, foreskin, face, lip, and scalp. We're going to do single-cell, multi-ohm ATAC-seq, and nuclear gene expression. We're going to do some spatial transcriptomics and also some single-cell RNA sequencing. And the idea is that we'll develop an atlas of pediatric skin development, which will bridge the developmental atlas and the adult skin atlas, and improve understanding of cellular differentiation and biology of disease, and give contextual insights across the human lifespan. I think it's done a little stop. There's one slide before that. Okay, so the skin structure. So basically, the skin has an upper part called the epidermis, which contains keratinocytes, and a lower part called the dermis. And underneath the dermis is fat. In the dermis, you've got blood vessels and nerves and immune cells, but you've also got these complex structures called appendageal structures. So in hair-bearing skin, you've got the hair follicle, which is here, and you've also got the sebaceous gland, which is this light brown area there. You've also got sweat glands. There are two types of sweat glands. Eccrine glands are present in most places, and there's a specialist type of sweat gland called the apocrine gland, which is present in hair-bearing skin in the groin, axilla, and scalp. In a bit more detail over here on the right, the epidermis made up of keratinocytes stratify to form a skin barrier. Skin barrier is really, really important. It's made up of cholesterol, ceramides, and is protective against everything. So it's protective against allergens getting into the skin. It's protective against us losing water and losing protein from our skin. You've got the all-important melanocyte, the pigment-producing cell in the basal epidermis, and you've got a variety of immune cells. Muzz led this project, which was published in Science, which showed that there were 34 cell types in the healthy adult skin, and at 7 to 10 weeks conception, there were just 14. So we're trying to work out, again, what's in between. So major questions are, what happens to infantile epidermis in the first year of life? So we know that the skin barrier is very weak when the baby is born, but that improves over the first year. We want to learn more about what happens to the sebaceous gland in the neonatal period and during puberty. We want to know more about changes in cutaneous blood vessels, especially in the first year of life, and I'll explain why shortly. And we also want to know about changes in the pediatric skin immune composition and function over time. And we're also very interested, because of our cohort, in differences in skin of color. So there are some known differences in normal skin physiology in skin of color. We know that people with different skin colors produce different types of melanin. We know that there's a different distribution of melanosomes, which are the pigment-containing organelles within the keratinocytes. We know that there are differences in hair shaft shape and hair texture. We know that there are differences in the size and activity of fibroblasts in the upper dermis, particularly in black skin. And we also know that there are differences in apocrine gland secretion. Over to your right, you've got this electron micrograph, which shows the melanosomes in black skin. You can see that they're quite large and they're single, so they occur on their own. In white skin, in contrast, they occur in clusters. There are many examples of skin disorders that are more common in skin of color. So for example, dermal melanocytosis, this picture here on the left is a blue mark, which is quite common in Asian and black infants. This is caused by melanocytes going down into the dermis. So what makes those melanocytes go down into the dermis? Tinea capitis, which is a fungal infection that affects the scalp in all types of skin, but is generally more severe in black patients. So there's something going on in the hair follicle. Keloid scarring, over here on the right. In black skin, you can get much more severe keloid scarring. Is this because of activated fibroblasts? Some people have shown possibly it may be related to changes in Schwann cells that surround the keloid. Atopic eczema occurs in up to a quarter of children. Probably some of you have had atopic eczema. In the baby, so in the infant, it's generally really bad on the face. In the older child, it's worse in the flexures. Why is that? We don't know. In skin of colour, the presentation of atopic eczema can be very different. So you can get this attraction to the hair follicle on the left in black skin, and you can get massive depigmentation or loss of pigmentation in darker skin as well. Acne can occur at different ages. We all know about acne as a teenager, but acne can also occur in the neonatal period, in the first few weeks of life, and it can also occur in the infant. This graph just shows DHEAS, which is an adrenal androgen, and how it changes during life. Pink is female, male is blue, purple is both, and you can see basically there's a peak in early life in the first few weeks, then it comes down, and then it goes gradually up, peaking after puberty. Benign tumors of blood vessels. So this is why we want to know about blood vessels. So on your left here, the first one is a capillary vascular malformation or port wine stain. These are present at birth and persist. In the middle is a rapidly involuting congenital hemangioma. This is something that grows in the womb and then involutes in size from birth. And the last one is the common infantile hemangioma or strawberry hemangioma, which generally is visible at age 4 to 6 weeks, but it proliferates between 4 months and 9 months and then regresses, as you can see in the picture. Autoimmune skin disorders such as alopecia areata and vitiligo occur between the age of 4 and 8 years in general. They start then. They don't start before that. Why is that? Is it changes in the hair follicle immune privilege, changes in the immune composition? Then there are rare genetic skin disorders. So a rare genetic disorder occurs in less than 1 in 2,000 of the population. 8% of the population, if you look at all genetic disorders, have a rare genetic disorder. So they are actually quite common. There are over 600 rare genetic skin disorders for which a gene is known. They normally present in the first few years of life, but can present up to the second and third decade. This is one example, the ichthyoses. So ichthyosis means scaly skin. And this is a collodion baby on the left. So the baby is born red, covered in a membrane of thickened skin. This falls off, and then the skin is scaly and red, like the picture in the right. There are over 40 different types, and it's due to a genetic defect in the skin barrier. So there are 40 known genes at the moment. There are probably about 20% to be found. So certainly knowing more about gene expression in skin and in the skin barrier will give insights into this disorder. Finally, just to mention epidermolysis bullosa. This is a blistering disorder which presents from birth. It's caused by mutations in genes encoding basement membrane components. The basement membrane is a 30 nanometer thick, so it's quite a narrow area, separating the epidermis from the dermis. This is a very severe disorder. Patients get continued inflammation, scarring, and then they develop skin cancer and generally die in their 40s. So moving on to public engagement. As part of this project, we're going to use the Centre of the Cell, which is a unique science learning centre within our institute. This was the first in the world to be actually located in a research lab. I think there have been more since. There's content from Queen Mary Science and scientists, and there's strong involvement with the local community with staff and student explainers. There have been over 200,000 participants to date. And the idea basically is to increase interest in science and in medicine, in children in the local community, and to encourage them to aspire to careers in those areas. So we hold educational shows and workshops. There's interactive learning in something called the STEM pod, which I'll show you. There is a youth membership scheme. These teenagers meet and discuss ideas, discuss educational shows and workshops, give input. There are also evening events. We work with children from age 4 upwards, school, family, public and community groups, and other partners. This is the pod. So the pod has got a nucleus inside, which is this big ball here. And that basically opens up, and there are lots of interactive games. At the moment, this is being completely refurbished with new games. And here you see some children in the pod. So for the Human Pediatric Skin Atlas, we're working on a plan which involves 3 parts. The first part is a participant focus group. So this is a group of kids here who are working on a heart, and you can see you've got a platelet, you've got white cells, you've got fibrin. So we plan to have something similar for skin. We plan to have a science workshop show on skin on campus and in schools, which is transportable. So there is one at the moment called Snot, Sick and Scabs, which is about like snotty noses and smelly feet, stuff like that. And the idea is that the show will be transportable, so we could put it in a box and it could go up to Newcastle or it could go to the Sanger. And there's also a plan for a game for the pod in the centre of the cell, which we hope also to make available online, which will increase its global outreach. So I'd just like to thank CZI for funding, the rest of the Pediatric Skin Atlas team, the centre of the cell, my patients, and the patients and families who are generously donating their skin to this project. Thank you.
Yeah, thank you very much. And also for congratulations for your public engagement. It's really awesome. I was wondering whether you have, regarding your donors, if you have any biochemical data of the donors. And I'm telling you this because some of the diseases, not diseases, but conditions that you presented, such as acne or alopecia, they're androgen dependent, for instance. So it would be really nice to correlate, for instance, testosterone levels in those donors together with their transcriptomic data sets, and this applies for...
That's an interesting point. And actually the keratinocytes in the skin produce steroids and androgens. Mm-hmm. So you can actually learn a lot from the gene expression of epithelial cells. So we've no plans to do any other interventions like take bloods.
So you're not measuring androgens in blood from this
no,
question?
No.
Okay,
No.
Thank you.
And our next speaker is Jian Shu from Harvard Medical School. Please.
Can I get my slide? Okay, so my name is Jian. I'm a new assistant professor at Harvard Medical School. So today I'm gonna share some of you of the work that we did during my postdoc and also in my lab at Harvard University. So because I'm new, and so I wanted to thank all my wonderful mentors in the past who supported me during my PhD and postdoc. So and also close collaborator Aviv. And also, especially, I wanted to give my biggest thank to the team because I started my lab last year, and then they were all brave to join my lab and take the risk to join like junior faculty. And then also the funding source to support some of the research that I will be talking about today. So today I think we're going to share three short stories. And the first one is about how we can actually think about time. And then the second one is how we can actually think about space. And then the third is how we can actually combine this different information to study the placenta development. So the first short story would be related to how can we reconstruct the time information. So some of the people in the audience and also the speakers, they all talked about different approaches to reconstruct time information because it's a fundamental challenge. It's still not just in experimental biology, also in the computational biology community. If you really wanted to think about the Waddington's landscape and then wanted to reconstruct the whole landscape through single-cell snapshot measurement, and because there are actually no general experimental approaches that will allow us to watch a cell over time without sacrificing the cell if we really wanted to get the genomics information from those cells. So we asked the question, can we just reconstruct this computationally, because lots of other pioneer work also try to tackle the same problems. As you might understand that if you show these videos, that it's videos are also actually just snapshots, but they have a way to actually connect those snapshots. So that's like a simple version of how we can think about the time information and the time trajectories. But in single-cell biology, it's more complicated, because there are a lot of new computational technologies that will allow us to actually reconstruct the trajectories. But in our case, that we were wondering, can we actually think about the trajectory more like a probabilistic distribution change over time? So the model is actually to model each gene expression, single-cell gene expression profile as just a sample from a probabilistic distribution. And then the goal here is actually just to connect those distribution change over time. And I think Donna and other speakers also mentioned some of this pioneer work to really tackle this question. And then we use a slightly different approach that is inspired by a French mathematician, which I think is a perfect time and a space, location for me to really talk about this work. So the goal is to actually map the distribution changes over time. But how to do this? The method that we use is called the optimal transport, which was proposed by the French mathematician Gaspard Monge. And if you have the chance, then you can actually visit his tomb. It's actually in the center of Paris, where he was buried together with other great scientists and artists in France. So Gaspard Monge proposed the original optimal transport concept during 70s, 80s. But in the original optimal transport, that their goal is to move a pile of dirt into different locations because they were trying to build the fortifications during the wartime to help Napoleon's army in campaign in Egypt. So, they need to figure out a way to move this dirt in the most efficient way in different locations. So we think, oh, this seems very similar during the development processes. We wanted to move a pile of cells into different locations. How do we actually do that? But there's one difference between the original optimal transport and then the cells, because the cells do grow and they die as well. So the probability, the distribution of the cell will change, because if the cells grow faster, then they will have more descendants. If the cell died, and then the population will be missing from the distribution. So we have to somehow optimize this original optimal transport method to add the proliferation rate and the death rate into the original model, so that we can actually better reflect the biological processes. So that was the basic fundamental mathematical model to really think about the trajectory. And we worked with software engineers in Aviv's lab and developed this method called the Waddington OT with this software that you can do many, many things. And the first is actually you can actually visualize a very large-scale single-cell sequencing data, and then you can use this to reconstruct the trajectories, predict the transcription factors or gene regulatory work along each of these trajectories, and infer also cell intrinsic ligand receptor interactions, as well as identify very large chromosome abbreviations from RNA-seq data. So we apply this Waddington OT to one of our favorite model, which is the iPSL reprogramming model, and we generated the single-cell RNA-seq data from 18 days reprogramming process. So we collect samples every 6 to 12 hours, and then we did this in 2 different conditions. One is called the 2I condition, and then the second one is called the serum condition. And those are 2 popular conditions or medias for growing iPS cells. And we use the Waddington OT to reconstruct the whole developmental trajectories of the reprogramming process. And as you can see that these are the two conditions. One is the serum, and then the second one is the 2I. And interestingly, that in the serum condition, we found lots of heterogeneity that we didn't expect. For example, we found the placenta-like cells here, and also neuronal-like cells, stromal cells as well. So I think this is the beauty of single-cell sequencing, because previously in the reprogramming field, through bulk analysis, we didn't realize that those cells which didn't succeed to be reprogrammed to iPS cells actually are a lot heterogeneous. And then if we just zoom in the placenta-like cells or the trophoblast-like cells, we actually see a lot of heterogeneity in the trophoblast-like cells, and then you can see lots of cells, they show different signatures of different subtypes of placenta or trophoblast-like cells. So that's why I think this is interesting data sets, and lots of the people in now the longevity research that they're also trying to use a similar approach using partial reprogramming plus single-cell RNA-seq or single-cell analysis, trying to figure out how we can actually rejuvenate or expand the lifespan. So with the Waddington OT, I think the most useful application for the Waddington OT is actually just to predict the trajectories. So we can use this to predict the past and the future cell states. So this shows you that if we look at the ancestors of iPS cells, and then you can see that the cells actually follow a very narrow string from the iPS cell stage, from the MET stage all the way to the iPS cell. So this is a reverse time. So we just wanted to see what are the ancestors of the iPS cells. So in summary, the first short story talks about how we can use optimal transport to reconstruct the time information. And then we developed this method called the Waddington OT, and there are some other methods we use similar approaches. And I think there's a large variety of different methods, and then I think it all depends on the biological question that you're asking and what kind of approaches might be more appropriate. And then we apply this to study the iPSL reprogramming. And in the following up studies now in my lab, we're asking the questions, can we identify, for example, the shared mechanism of different reprogramming strategies, the chemical reprogramming, reprogramming by lineage specifiers, or other reprogramming cocktails? And we're also asking the questions, can we actually identify the very early stage events during the reprogramming process? And then you can use this to do this partial reprogramming. And I think for the last one would be something that in the computational biology community might be more interested in, is how can we actually leverage the large single-cell sequencing data from the human cell atlas to generate combinatorial predictions of how can we actually make different cell types. So what kind of transcription factors might be helpful for the reprogramming. So that's a very short first story about the Waddington OT. And then the second one is how we can think about space information there. Because if you look at the single-cell genomics data, usually you don't have the space information. That's why we're thinking about are actually very fundamental questions. And then I think I also wanted to ask the audience that how do we actually think about to define the cell states, cell types, or phenotypes? Because previously, I think originally cells were actually discovered by imaging through microscopy in 1665. And then with the more recent advent in the single-cell genomics field, that we started to annotate or define the cell states or cell type or cell phenotypes through single-cell genomics. However, I think both methods have pros and cons. For example, imaging can be very, very low cost and non-destructive and high throughput. But the problem for imaging is that most of these imaging features are not interpretable, so we don't know what does it mean with all these features. At the same time, genomics is very, very powerful, and then also one of the biggest advantages of doing genomics is because it's interpretable, because we know what does it mean with each of these features, those genes of this after you sequence them. But the challenges for genomics is that it's destructive, so you have to kill the cells to get the information. If you kill the cells, which you will not be able to see the same cell in the future state or in the next time point. And also, genomics has very limited throughput. That even with this massive scalable single cell, RNA-seq or single cell multiomics, still it's not at the throughput that we can really apply this in the large population scale. So that's why we're thinking about, can we actually leverage the best part of these two worlds and then just combine the imaging with the omics information through machine learning? It's actually a very exciting time, I think, for this type of work because of the technology foundations, especially in machine learning in recent years. They actually revolutionized lots of this work, not just in computer vision or in biology or drug discovery. For example, ImageNet curated or developed by Dr. Feifei Li and others at the Stanford University, that they collect all these large collections of different images, and then they teach computers to recognize what is a cat, what is a dog, right? And then they wanted to translate the imaging information into labels. And then more recently, I think by DeepMind, AlphaFold, they're also doing something very similar, but not exactly the same. But we wanted to translate the sequence into structures. And then even more recently, I think in the pop news and social media, people might have seen this, that for example, OpenAI, they have this very cool model that you can actually translate text into images or even to videos. But all of them share something in common. So they wanted to translate different information or multimodal data translation. So wanted to translate one data modality to another data modality. And then these different data modalities are actually describing the same or similar information. So we ask the same question. So can we actually think about how can we translate the imaging data into the genomics data? It sounds intuitive, but it's actually very, very difficult, because the challenge is that genomics data or omics data are super high dimensional. That's why we wanted to think about as an input as the imaging data. We also probably wanted to use very high dimensional imaging data. That's why we started to look at what kind of imaging methods are high dimensional. So that's one of the methods that we wanted to use, which is called the hyperspectral imaging, chemical imaging. It's a super high dimensional imaging. So each pixel is 1,000 dimensions. So think about this as 1,000 colors. And it's a subcellular resolution. So it would be a high dimensional imaging data. And then the question would be that if we use this high dimensional imaging data, can we actually infer or predict the single cell omics data? And then in the future, maybe we have enough data, then we don't need to do omics or sequencing. So this is a wonderful, wonderful collaboration with Aviv. And also it was because one of her super talented postdoc, Kosaki, he was trained in the Raman microscopy field. And then after we published the Waddington OT paper, and we started to chat, because we realized the challenges of using computational trajectory inference methods through single cell sequencing. So we asked, can we actually combine his favorite Raman microscopy with our single-cell sequencing data during the reprogramming process? And then this is also in collaboration with Peter So at MIT, who is an expert on Raman microscopy. So we developed a method called the Raman to RNA, it leverages the rich information that we can get from the Raman microscopy. So what is the Raman microscopy? So it's a chemical imaging method that measures all the chemical vibration inside of a cell. So it's a composite measurement of all the chemical reactions inside of a cell. So it includes lipid, protein, nucleic acid, and all the other molecules. So I would say this might be like a pseudo multimodal, true multimodal measurement, because it measures completely different things, including lipid and protein and others, but it's not interpretable. So you will get a spectrum like this, and then some of the peaks we know what they are, but most of them we don't know what they are. It's similar to when first we sequence the human genome, we don't actually understand what does it mean with all those gene names, and it's the same problem here. So we ask the questions, then can we actually try to infer this spectrum data to the single cell gene expression data? So that's why we work together on this Raman to RNA methods that we did a very simple experiment in the beginning. So we mixed the fibroblast together with iPS cells, and then we did the Raman images and the single cell RNA-seq, and then also using some single molecule fish and to map some of these marker genes as the anchor to link the Raman microscopy data with the single cell RNA-seq data. So this shows you that you can actually do a pretty good job to distinguish what is the fibroblast and what is the iPS cells, and then use some of the generative models to generate a single cell RNA-seq profile from the Raman images. This shows you that if you just look at the Leuven-Castorines, this is purely just the Raman data, and then each individual genes that you can predict it from the Raman spectrum data. It shows pretty good results. And then we asked, can we actually demonstrate this in a time series data? Then you can actually watch your cell over time. And then from these movies, then you can infer the single cell gene expression. And so we apply this Raman to RNA to the same system that we showed before you during the Waddington OT paper, that we did imaging Raman images, and then we did some single molecule fish to map some of these marker genes, and then we use single molecule fish data as an anchor to link them with single cell RNA-seq data. But that was an initial test, initial trial, proof of principle. There's so much room to improve, and then I think there's no need to. It's possible to do this without the anchors as well. That's something that we're working on now. And this is just one very simple summary slide to show you what this might look like. So the blue dot shows the reference, the ground truth single cell RNA-seq data that we generated from the Waddington OT paper, and then the yellow dots shows the generated the single cell RNA-seq data generated from the Raman spectrum. And then if you plot them on the UMAP space, then you can see that they overlap with each other pretty well. And then the differences between these two different data sets is because from the single cell RNA-seq, we have a very, very large data sets, 250,000 cells. But with the Raman, we have way fewer cells. So that's why you see it's more sparse. And then on the right, that you can see that if we just look at the gene signatures of iPS cells, so the nanog, octafor, or those gene modules together, and then if we just predict them, the gene expression, and then you can see where they are on the UMAP space. So in summary, that we tested this with the Raman microscopy, but I think that's just like an overall example that how we think about this doing tissue mapping, that genomics is great, and then it's a wonderful and useful information. But in addition to that, there's all other features that usually we don't measure, or we don't take into consideration when we think about the cell representation, like cell states. And that's why we started to test this idea, how we combine different kinds of imaging data with different kinds of omics information. So this shows you some of these imaging methods, like label-free imaging methods that's very fast and scalable. For example, from the left panel, that with this infrared imaging methods, that you can actually scan the whole mouse brain slice within 9 minutes. So that I think it is one of the advantages of thinking about all this label-free imaging. And then on the right, that's a 3D imaging methods that measures the reflection of light. So in summary, I think Raman to RNA demonstrated the first live cell predictions of genome-wide RNA expression from single cell Raman microscopy data. And then I think this lays an overall general principle, like a foundations or frameworks, how we think about translating different kinds of biological data between different data modalities. And then intuitively, then we're asking the next question would be, can we actually predict some other single cell information, such as single cell ataxic, single cell proteomics? Can we demonstrate it in large tissues? And can we do this in time-lapse imaging? And then lastly, just one slide to show this, that we are actually working on this with applying all these technologies to study the placenta in mouse, marmoset, monkey, and human. So, and I know that in the audience, people are also very excited about this organ, and I think there will be lots of synergies to really think about using different approaches to study some of these organs. So, in last, that I wanted to give these questions to the audience, and then we can all think together whether it makes sense to start to think about whether we can think about like a Google Translate for biology, that you can translate different kinds of information with each other. And then we'll thank my lab members, and then they are also the true people who actually did all this work. And then lastly, we're actively hiring. So if you're interested in joining us in Boston, just let me know. Thank you.
Thank you, very impressive. Would the second model, the Raman, would it work, or did you challenge with mutants, for example, something like a mutant of actin that would change the structure in a way that might not be able to recognize anymore a fibroblast as a fibroblast?
Yeah, yeah, that's a wonderful question. We didn't show in the, we didn't put it in the bioarchive, but we did a very quick and dirty follow-up studies with Rudolf Janishet Whitehead. He has actually a isogenic iPS cell lines with 1 point mutation. So you have the healthy wild type as a control, and then we mutated one nucleotide, and then we did the Raman microscopy, we did Raman images. Actually, we can find very drastic differences if you just like make a 1 point mutation. However, it's actually very difficult to find these differences through single-cell RNA-seq. So I think this could be very useful complementary informations in addition to the omics profiling.
So I wanted to follow up on the Raman microscopy as well. You know, one challenge, of course, is that the vectors that you measure aren't interpretable. But a more fundamental question is, do they contain the information? So if you run Raman microscopy on a complex tissue, and you run something like a TISNE or a UMAP on the result, does it generally look like it contains the information of the cell types in the tissue?
Yes, yes, that's also a wonderful question. So if you just look at the Raman microscopy data, and then you just did the UMAP or like a very simple classification or like clustering, you can actually identify the overall structures of different cell populations. And for example, like the mouse brain data that we show, it's not Raman, but it's another similar approach. That's just purely based on the imaging features. And then if you look at that with the Murphy data, it seems like pretty similar. And then that basically gives us the confidence, or like at least the hope that maybe it is actually possible to translate these different data modalities, as long as they share some overall structure, the distribution shares some structures and some similarities. And another, I think, I would like to add that, like, maybe one imaging method is not enough. And if you think about all the features that represent the cell state, omics, or RNA-seq, or ataxic, or even methylation, or morphology, chemical vibration, those are all just a small portion of all the feature across all the feature space, right? So I think if we just think about by combining orthogonal, completely different features, and then maybe that will help us to better annotate or like understand the cell states, and in addition to just single cell multi-omics, because there's information redundancy if you just adding all kinds of omics information, because it's always in the similar cloud of feature space. So yeah.
Yeah. Okay. Thanks.
If I may, it's around this aspect where what you were trying to show is the possibility to translate the imaging data in omics-related information. But what you're actually saying, which I find even more interesting, is to go beyond, is to enrich the overall information by using the two of them. And so maybe you can elaborate on that. And then the other question is, can you use, because you are putting forward the label-free imaging, so have you tried anything with ultrasound images or any other type of imaging that is used commonly in the medical area? Because that could be extremely useful to have non-invasive approaches.
Yeah, and that's also a very wonderful question. So I think the first question is, I think it is always useful to combine, integrate different features or different data modalities and combining images with omics, and I think that's very, very useful for annotating the cell states. But I think like the point that we're trying to make here is actually, I think many people are actually doing this integration. But with this development of generative models, and then maybe we can think about this translation. But the application we are trying to see here is, because genomics informations are difficult to generate, so whatever measurement that's easy and ideally non-destructive or cheap, so then you can use those easy, cheap, and simple measurement, or even low-dimension measurement to infer very difficult to generate data, such as those like very high-dimensional data. And so I think the two general direction would be from low dimension to high dimension, and also from low resolution to high resolution. And then it's more like efficient mapping, I would call it that way. And then the second question was, have we actually tried some other imaging methods? One of the undergrad students who's actually here, Charles, and the second people here in the photo there, he developed some methods using HE staining to predict single-cell RNA-seq data from HE staining, which is more applicable in the medical settings. And he's now a PhD student in my lab. He did that work together with Avi when he was with a undergrad student. So I'm just saying that we're actually actively looking at other imaging methods, maybe like low-resolution MRIs, and then HE staining, bright field. If you just combine all of them, not all of them, just combine like the cheaper ones, and can we actually use this to predict like multi-omics data? And it will not be exactly perfect, but I think it is an overall goal.
All right, thank you. Thanks again.
Thank you.
And moving on, our next speaker is Edouard Hannezo from the Institute of Science, Austria.
So thank you very much for the invitation. So I'm a theory lab, so we're a theoretical biophysics group, but we work on a number of developmental problems in a number of systems. So actually what I'll show you today is mainly some collaborations on vertebrate, in particular mouse early embryogenesis, but we think that many of the theoretical tools that we're thinking about are quite broadly applicable. And in particular, everything is going to be around sort of using topological information on the cell contact and the cell context to understand how embryos behave. So one big question, one big topic that has emerged, I think, in the past 10 years in developmental biology is that, of course, both sort of shape and patterns are emerging. So the shape of the embryo is emerging at the same time as the cell fates are being specified. And it's really not some unidirectional things, right, where the cell fates come first and then give rise to shape or vice versa. It's really a process where the geometry, the fate acquisition and the mechanical properties sort of constantly feed back on each other. There's a really strong dynamical feature to all of this. And in particular, one thing I wanted to talk about is we're quite used to think about cell fate transitions, the idea that you can abruptly go from, let's say, or bifurcate from some cells to some cell fate types to others. But actually, we found that this happens also in terms of mechanical properties, that mechanical forces, mechanical properties of tissues can change extremely abruptly, and that this is really important to understand development. And in particular, so in general, we want to understand here how are these sort of changes in mechanical properties affected and coordinated with changes in cell fate. So one first question actually that sort of intersects a bit the previous talk is often in one, I'm going to just show you two slides on something that is published, and then because this is the basis for some unpublished work that I would like to share. So one of the core ideas is that, of course, mechanics is hard to measure, right? And ideally, we would want to have some image-based methods where basically we can look at an embryo and extract information on its mechanical properties. And so in something like zebrafish, for instance, which is some work done with the lab of C.P. Eisenberg, these are 3 snapshots of zebrafish gastrulation, sort of early of a tissue. So here you see cells and their contact with the white being the extracellular fluid. And these are 3 time steps of development. And when you measure actually directly the mechanical properties, you see that it undergoes an extremely sharp transition, which is key for gastrulation. But if you look at these pictures, you know, if I would ask you to guess which one is completely different mechanically from the other 2, it's not so obvious, right? Sort of, if we actually do the measurements, right, it turns out that it's the middle one, which is fluid, but at least it was not super obvious to me looking at these 3 pictures that this was a completely different mechanical object than the other 2. And so we thought quite a bit about this, and it turned out that actually one way to think about this is is to understand the cell context, which cell connects to which other cells. And it's actually something that in sort of physics of granular materials is something that is quite common. It's useful not to think about cells, but instead cell connectivities. So what we do often is we take these embryos and we make them into graphs where a node here is a cell, and there's a bar here to tell a cell contact. And this tells you about the cell connectivity, which cells are connected to which cells. And why is that useful? Well, we know actually from Maxwell, from the end of the middle of the 19th century, that actually the mechanical properties of objects can be drastically dependent on connectivities. If you take a bridge, for instance, a bridge, after all, is a succession of these sort of links here, which are exactly similar from a physical perspective to the embryo connectivity map I've showed you. And here, for instance, this is a bridge. You can see that this is bearing load, right? In principle, if a train can go on top, because it's resistant to deformation, right? You have to expend energy to deform it. But if now, for instance, I remove a single link, right, to my bridge, you can imagine that there's going to be something pretty dramatic, right? Removing a single link in my connectivity is going to completely change the mechanical properties, and we think that this is exactly what's going on with the embryos. The easier way you can see about this is if you think about 4 cells, for instance, that have like 4 connections between the 2, you can pull on one and it doesn't cost any energy because you essentially can deform the 4-cell configuration without sort of straining any bond. And so that's, from a topological perspective, something floppy, whereas if you add a single bond on top of that, suddenly now you have 5 bonds and 4 cells, and this is rigid. This is something that has some mechanical rigidity. The advantage is that this is a purely topological criteria. You can look at images, and from this criteria, you can say what should be solid and what should be fluid in an embryo. Okay, fine. So does it work? So again, this is published, actually, so I'll go pretty fast. But the key insight from Maxwell is if each cell is connected to at least 4 neighbors, if it holds 4 neighbors, more than that, you're going to have something solid. The embryo is going to stay put and not deform. Whereas if it has less than 4 cells, suddenly it becomes extremely fluid. It's very prone for deformation. And this is a graph basically that summarizes over many, many different perturbations on fate, mechanics, wild type, different time, different regions of space. You can correlate extremely well the mechanical property of an embryo to just this sort of image-based criteria of its connectivity. So the baseline this is published, and so this is just to say that sort of connectivities, knowing which cell is connected to which cell, is a really, really simple image-based criteria that tells you a lot about how embryos how the mechanics of embryos and how embryos deform. And you can go one step beyond. You can understand, okay, how are these contacts? How are these contacts organized? And again, we've known quite a bit in the past few years. On the physics of cell-cell contacts, in particular, if you take 2 cells, for instance, they can change their tensions. For instance, here, so they can regulate their cadherin to increase the sizes of their contacts, which is very analogous actually to the physics of soap bubbles. And if you take, for instance, 4 cells, for instance, which have here on the left very few contact, very little adhesiveness. They have very little compaction. Well, you can see that they're actually in a floppy configuration, so you can pull on them and it's not going to cost any energy, so the embryo is going to deform very easily. And as soon as you increase your adhesion by a tiny bit, there's a critical threshold of adhesion here in this dashed line, where suddenly you make some extra bonds, and suddenly your configuration became rigid, and suddenly you can't sort of deform your embryo that easily. And so again, this is just to give an insight that relatively small mechanical changes, for instance, by how compact is an embryo, how cells regulate their contact, can have dramatic impact on their connectivity and their mechanical properties. Okay, so this is essentially a long preamble to tell you about a completely ongoing story, actually, with the lab of Takashi Iragi on the early mouse embryo. And this is the postdoc work of Dimitri Fabrege in particular. The theory was done in my lab by Bernat, and all of the computational analysis by the group of Virginie Ullmann in EMBL. And the question, basically, is to say, here, what I've shown you before, we were looking at pretty large tissues in gastrulation. We had thousands of cells, and so we were looking at average connectivities. But there's a pretty interesting question which turns out, which is to try to understand the connectivities of cells during early embryogenesis. For instance, in particular, early packing, so that the 8-cell stage in the embryo, are there some specific packings? Do embryos look like all look the same? Do they connect in the same way to other neighbors? And is that important for morphogenesis? This is one of the questions we wanted to ask here. And in general, by the way, as a last slide of introduction, this is something that people are more and more interested in with advances in sort of imaging and 4D imaging and tracking, where we have more and more information on the packings and on the quantitative features of early embryos, but often we don't really know still what to do with all this complex imaging data. So let me cut this. So the pipeline that sort of the pipeline that Dimitri developed essentially is to take. So these are actually mouse embryo, but he's been looking at this also for rabbits, for instance, and monkeys. So the data set essentially is to look in live and to segment entirely the entire time course of a cell of an embryo from a single cell to essentially 16 cells. And so all of these embryos are then, it's around a data set of 30 embryos. They're all segmented, they're all tracked. So we have all of the membranes, all of the angles, all of the trackings, the cell movements, etc. So it's a bit of it's a dream data set for theorists, because from this, we should really be able to understand, first of all, are there some, what should an embryo look like? Are there some stereotypical features that a mouse embryo should look like? But also, are there some interesting theoretical predictions we can make out of this? So what do we do? What we did with this? So the first thing we did is to look at from first to 16 cell stage, and we looked in particular, do cleavages have a role in arriving to a stereotypical shape of an embryo, for instance? And there it was a bit of a mess. I mean, the cleavages were really non-invariant. There was no correction, so there were very little structures to this data. So then we looked at something even simpler. We looked at the 8-cell stage, and we asked, throughout the 8-cell stage of a mouse embryo, do they converge towards a well-defined packing? Do they look more? Do they look more like each other at the end and at the beginning of the 8-cell stage? Can we essentially make an atlas of cell shapes in early mouse embryo? And so the first thing that we did essentially is to take all of this segmentation, so essentially reconstructed splines of all of these cells. We have all this connectivity, and to try to make an unbiased classification, essentially a morphomap, a dimensionality reduction to see whether the type of shapes that you should expect from an embryo. And so this is the first outlook. So here, one thing here is a trajectory, because we have all of this information in time. So these are 30 trajectories of mouse embryos throughout the 8-cell stage in a low-dimensional representation. And one of the interesting things, and we tried it in many different ways, one of the interesting things essentially is if you look at the distance between different embryos, they get more similar to each other as a function of time. Early on in the 8-cell stage, they're very dissimilar, but if you look at the average distance, basically, they seem to converge towards some relatively specific structure. So essentially here, for instance, this attractor here seems to be special, cells seem to be attracted, so it seems somehow that there's some regulative mechanisms throughout development that tells the embryos that should look more like each other throughout this 8-cell stage. And so, of course, it's nice, but then there's a bit of a question. Of course, it's hard to interpret these kind of maps, right? What's special about this point right here? But the cool thing is that because these are physical systems, these are connectivities, the idea is that we could use topology to say something to guide us in this kind of high-dimensional space, and use sort of topologies and this kind of connectivity maps as some landmarks to understand a bit better intuitively where we are in these kind of things. And so in principle, for instance, if you go back to this, if you think about how to, we played actually, we literally took sort of 8 spheres and tried to pack them in different ways. There's of course an infinite way that you can pack sort of 8 balls together, right? This can give rise to many different shapes. But the cool thing is that most of these shapes are non-rigid, right? Most of these shapes, you can easily deform them, and therefore they're expected to be not very stable. If you ask now, what are the rigid ways you can pack 8 cells, there's only 13 of them, actually. This is something that has been looked at in the physics literature. And so the mechanically stable ways of packing 8 cells, there's only 13 of them, basically. So that's, I mean, they have names which is based on sort of symmetries. It's not important. But already, this is useful because suddenly you go from an infinite possible phase space of packings to only 13, right? So that gives you a guide already of say, we would expect that the embryo should go towards these 13 stereotypical shapes. This is a theoretical prediction. And then we ask, do they have the same, so they're all sort of rigid and stable, but do they have the same energy? And it turns out, yes, theoretically, if you take this kind of, in the physics literature, all these 13 configurations have the same energy. So in principle, the theoretical prediction from all of this is that we would expect embryos to not have an infinite amount of possibility, but converge in time towards these 13 possibilities. Is that true at all? So the first thing that we did is to look, just to test the basic assumptions of this, and in particular, what happens throughout time. I mean, there's 2 things that happen throughout time. One is that the embryo, the main thing is that the embryo compacts. And so if you look early on, for instance, if you look at the number of non-rigid, sort of this non-rigid structure, there's many of them. And as a function of time, suddenly, all of the embryos converge towards the 13 possibilities. Basically, there's no non-rigid embryos at the end of morphogenesis. So, and I'll just have 2 slides, I'll go a bit faster on this. You can actually look at how they converge. So this is an early 8-cell stage, and this is a late 8-cell stage. And you converge towards VHE, and that's really because you compacted, and therefore you were able to make more contacts and go towards something. This is something that theoretically we can explain quite well. So we did image-based modeling. We take an early embryo, and we model compaction in a really minimal model, and we can explain very well this kind of convergence towards a specific shape. And the last thing I want to give you with is that, so we predict that suddenly you don't have an infinite amount of possible embryo shapes, you have only 13s, but actually the surprising feature that actually, we thought initially this 13 would be equal, but actually it turns out that there's only a few that win. Out of these 13 packings that you could expect that are stable, the embryo actually converges towards only 1 or 2. And this is something that is even more surprising to us, but actually we did some more theory, and it turned out that actually this, if you now theoretically compact embryos, it turns out that upon compaction, compaction makes some structures a bit more stable than others, and it predicts in particular that sort of one structure should emerge from from a purely physical reason upon compaction, and which turns out to be actually the experimental one, the experimentally selected one. So this is again completely still very much preliminary, but this is just to say that based on really, really simple topological classification, we can make sense of the kind of structures that the mouse embryo looks like, and from physical consideration, we can explain why they seem to all converge towards 1 or 2. And that's the end of the talk. I'd like to acknowledge in particular our collaborators, Dimitri Fabrej, a postdoc in the lab of Takashi Hiragi, my group, and thank you for your attention.
Thank you. Question up here.
So I've always been fascinated with shape and movement in embryology, but you know, we understand cell fate in terms of their gene regulation, chromatin, transcription factors. Sure, there's cytoskeleton stuff, but what is the sort of mechanism that creates the sort of converged shape? What are the forces that I mean, you've convinced me that it's a good thing to reach that state and that it's an ideal state to be, but what are the actual mechanisms that ensure the embryo does that?
Yeah. So this is a very good question. So the core mechanism, essentially, the one that gets selected is initially you have embryos which have very strange shape, and in particular, very anisotropic shape. So there are some that have the sort of a bit sort of sugar shape. I mean, there's some which are very non-round and symmetric, and the one that gets selected is the one that is essentially the more spherical, the more round at the end of the 8-cell stage. And one of the things that we're interested in this is that, of course, one of the ideas about the subsequent phase choices at 16 is that this is also position dependent, right? Sort of outside, inside is going to change the fraction of phase choices. And if you have something, for instance, that's very spherical, you're going to have a certain amount of cells that are inside versus outside. And if you make something that's very irregular, sometimes you're going to have only outside cells. In some of our weird initial configurations, if the embryos would stay like this, at the 16-cell stage, they would have no inside cell, basically. And so actually, Dimitri has been perturbing in particular cell synchrony to get embryos with weird shapes at the 16-cell stage, and he found defects in the fate allocation, basically, in looking at SOX2, essentially subsequently. So there is this idea that if you make your cell choices partially, at least on spatial information, having weird shapes is going to translate into different cell ratios, basically. And so we think this might, but again, this is ongoing, but it has to do with sort of scaling of numbers, maybe.
All right, thanks. We're going to have to move on.
Okay,
thank you.
Thank you.
And our next speaker is Leon Peshkin from Harvard University. Please.
Thanks very much for this opportunity to present. I'm really enjoying this meeting. I think this is going to be a different talk because I see that most people first do their work, then publish, and then come to give a talk. Here, I speak about a project which is very much in embryogenesis state, and I've come to ask you, what should I do with it, and how can I spot some of developmental problems to avoid issues later. So it's about a small sister project to Human Cell Atlas, Frog Cell Atlas, and I sort of speak as ambassador from Xenopus community. It's a lovely, closely knit community that got together about 4 years ago and decided that one highest priority for the community to move forward is to have single-cell atlas. And I have been sort of known to this community as data quality policeman. I always complain about things, and so they decided that the best idea is to task me with creating this atlas. We sent in an application, and thank you very much to NIH for having funded grant to create such atlas. Now, you might notice, if you're familiar with NIH mechanisms, that this is an unusual kind of a mechanism. They sort of gave me a dirty job, not to be very innovative, to be extremely careful and do things well. So it's about scaling up mechanisms, instruments which work very well to create careful data, useful to the wide range of research groups and institutes within NIH. The specific aims are about looking deeply at some of the embryonic stages and organs. Perhaps most important here is the third aim, to establish the pipeline. So I decided not to take a democratic approach to this, but more dictatorial approach, because I realized that the choice of research projects and methods is very much dictated for, at least for many of Xenopus biologists, but by not having availability of good bioinformatics, bioinformaticians and bioinformatic pipelines. So the way that we're trying to set this up is by having, we call it a research hotel. It's a place at the MBL where respective specialists who would like to profile normal development or normal state of developmental stages or organs, after they carefully debugged dissociation into healthy single cells in their lab, they could come and visit for a few days, and under the supervision or with help of our technicians, they dissociate organs sort of using same protocols and same quality standards. They barcode this, not using 10x, we're using in drop in order to gain a 10x factor in the number of things we can fund with this, taking into account the cost of barcoding and sequencing. Then the idea is that everything is sequenced to the same depth and analyzed in the same way. Since I'm sort of a refugee into development from machine learning, I very much care about data later being amendable to machine learning, yes, but also just reanalysis. We're also thinking of actually creating a boot camp where people come together and in one huge effort, we do everything in a very comparable way. So there has been a first atlas published about 5 years ago now, so it exists now. It's very nicely browsable. You can go and look at your favorite cell types and genes. It is not just single cell atlas of embryonic development. There are trajectories of development with cell states connected across time courses. It is also very nicely matched to respective zebrafish cell types. And I think this is one of the key challenges probably to make to make model organisms helpful to this community is to have standards of quick, effortless integration across species and across releases, because this effort was an effort of many years, careful effort of manually analyzing and matching every cell state. I want to remind you that we all love transcripts, but we're actually not made of transcripts. We're made out of proteins. Proteins are the functional units, or at least very important functional units. So I would like to talk to you, and I think a lot about mapping proteins and transcripts in this data. This is just one example of how we're using this. We made careful profile of protein expression. So the previously published atlas covered early stages 8 to 22. You could see that around stage 22, their extracellular protein being expressed and extracellular matrix being laid. So knowing exactly which proteins are expressed where naturally helps us select enzymes to do a nice to break them up to single cells. But I'm sure there are more creative ways to use that. We also develop guidelines for the whole community to dissect out the organs and perfuse blood in a very consistent way. We profiled proteomically tissues and organs to discover, to our great satisfaction, that most of the proteins actually nicely translate to human markers. There are a few interesting surprises, but I think surprises are actually less important here. We want to establish this mapping between human and frog markers to be able to go back and forth. So with those methods in place, I thought we're nicely set up to progress, and then a minor hiccup happened. Somebody completely scooped this whole effort. A couple of months ago, a team in China published a very nice paper covering all of the organs and embryonic stages in frog. I'm exaggerating. It is a very nice paper, but the whole thing is about 500,000 cells. So I take it as an opportunity to go and see what needs to be done, where we could focus and drill deeper, and particularly how to make this effort relevant to the Human Cell Atlas community. With this, I went to see what is the Human Cell Atlas community up to, and I saw this extremely disappointing figure. That's when I knew I have to come here, because for some reason Xenopus is unjustly penalized in every category. What's up with that? This is not right. So the rest of the slides in my talk sort of meant to give you a gestalt. I will not go very carefully through every slide. So first I want to point out that Xenopus is quite closer to human as a model organism and has heart, lungs, limbs, and fingers to model. Now, you may laugh, but there are genes which are important developmental and disorder genes which are missing in the rodent lineage. They are primate genes which are there in the Xenopus. This is one example, but there are actually groups of genes like that. Only in 36 hours you get an embryo with most of important lineages already in place. There is not one, but actually two species, and we always go back and forth between the species. They have respective advantages. One is convenient, excellent quality diploid genome. It develops faster, but it's smaller. The other one has much larger embryos, which are amenable to microsurgery and amazing manipulations. This is just an overview of an effort to map what was modeled in Xenopus in terms of human disease. So pretty much all major classes of human disease have been mapped and modeled in Xenopus. There is elaborate fate map, and so using this fate map, you can do amazing things. You can make a transgenic embryo, wait to the exact developmental stage where your tissue of interest is at a certain state, get a little chunk of this and graft it onto a normal embryo, and then wait to see what happens and characterize the effects. This is another very special way where you can use the left half of an embryo to control for the right half of an embryo. You manipulate only one half, and then this is a perfect control. So there are models for fundamental cell biology and cell behavior. Eye disease models, this is just some of the more, I would say, graphic examples of this modeling. Craniofacial syndromes, where you could carefully kind of stage by stage track what's going on. Most of these images and papers I mentioned have done very careful work of characterizing the disease and disorder, but did not use tools of single-cell transcriptomics. So imagine how much more we could do with that. So again, back to the cell fate, what you can also do is go and inject some drug. The volume is large enough to go with your bare hand and the needle and inject drug into a cell which you know has particular fate. There is a legend out there, I heard, that Xenopus is not good for imaging. Yes, there's actually yolk and pigment in Xenopus, but it quite rapidly disappears. So this is a beautiful image of, I think it's neurotubulin in Xenopus, just as an example. Where else could you make huge screens of toxicity? So you can, there are assays where you place embryos on a flatbed scanner. And there are regulations, of course, but you can justify this sort of experiments in Xenopus, where you look for drugs and toxins influencing the development. And again, I think there's a ripe ground for doing single-cell work once we establish the baseline of cell type distributions in these embryos. With this, I hope I convinced you that the next time the paper is published, you could make a correction for Xenopus, and I would be happy to discuss this. Thank you.
All right, questions? I think next time we'll make sure that all the dots there are absolutely equal in size.
I thank you for a wonderful talk. I saw the paper that you got scooped by. It looked like it was lacking thyroid gland tissue in their set of organs. Is that included in your atlas? Is a common model for thyroid gland to study toxicology in Xenopus, especially by the EPA in America.
So my atlas covers early embryonic stages for now. Okay. There is atlas published by another group. I cannot speak to that, but we are at early stages of planning. So come talk to me. Maybe we should include thyroid. And,
yeah, I think like stage 50 and onwards to like 60. I think a lot of people would be interested in that. Awesome. Thank you.
Hi, I was wondering what's the plans for dealing with the status of the genome in Xenopus levis? I mean, is that something that, if you're going to focus on that model rather than Tropicalis, what is the status of the genome at the moment, and will that be a problem for single cell analysis just because of its complicated genome?
So the state of Xenopus genome have been not so great historically. But today, and I believe for about a couple of years now, there's excellent genome. It hasn't really percolated to all the resources yet, but a Tropicalis genome, I would say, one of the best available genomes of all species at this point with gene models. And the Levis genome is pretty good. We're not focusing on one or another because there is a tradition and tools for going back and forth, and some of the things are obviously better done in Levis and others in Tropicalis.
Okay, so would the single cell atlas be in Levis then, or in in Tropicalis then? Or
so I anticipate that the Chinese published group is in Levis. Our embryonic is in Tropicalis. We will probably continue to do organs because of dissections and things like this. Mostly in Levis, but embryology in Tropicalis.
Can you cross-map onto the different genomes?
I'm sorry?
Can you cross-map to the different genomes? Have you tried that from like?
To the different version of genomes in the same species or across
so
species?
So if you have your cross-species mapping, like if you take
yes, cross-species mapping.
Put it onto Tropicalis, so to make it easier, I guess.
It's yes, the cross-species mapping is very well worked on and established. The Levis is pseudo-tetraploid, and the groups of genomes are available and yeah, straightforward.
Well, if there are no more questions, then thanks again. And that concludes the afternoon session. And now, and now we have a break, and then there will be breakout sessions. Actually, could you leave up the schedule for a second? Yeah, just so people see where they're, so here you can see where you're going for the different breakouts.